monitorava le capre, però mi faceva vedere tramite videochiamata, una tenerezza unica. Ditelo. Sì, certo. La presentazione è quella.
Hereby, I open this academic ceremony in which Elisa D'Alessandro will defend the academic thesis Atrial Fibrillation and Hypercoagulability, a two-way street with many side roads. May I invite you to present a summary of your study and the conclusions of your thesis?
Thank you, Prorector. Dear members of the Defense Committee, dear family and friends, dear colleagues, thank you all for being here today. Thank you also to those who are joining online. In the next 15 minutes, I will share with you some of the main findings of this thesis entitled Atrial Fibrillation and Hypercoagulability, a two-way street with many side roads. As you already know, this thesis is the result of a joint effort between two PhD candidates, myself and Billy, who will defend this thesis right after me. Many people would say that Billy and I are two very different persons, and in fact we are. However, during these years, we made use of our differences to work together, brainstorm, perform experiments, and finally to write this thesis. We spent so much time together that eventually, without knowing, we would come to work wearing the same clothes, just like in this picture here. Today, we are both proud to share with you some of the main findings of our joint effort. And so, let me start with a question for you. Which comes first, the chicken or the egg? I'm sure that at least once in your life you have wondered about this question and you were probably not able to answer it. In fact, for millennia, this has represented a philosophical dilemma. Nowadays, it is used when, between two events, it is not clear which should be considered the cause and which should be considered the effect. In this thesis, the two events are atrial fibrillation and hypercoagulability. Understanding the relationship between these two was one of the main goals of this thesis, and it's also the main topic of this presentation. Before giving you the answer to my question, I would like to tell you a bit more about atrial fibrillation. In this picture, you can see a cross-section of the heart. You can see that the heart is divided in 4 chambers. The upper chambers, which are the right atrium and the left atrium, and the lower chambers, which are the left ventricle and the right ventricle. The contraction of the heart is controlled by organized electrical signals that start from the right atrium, spread over the left atrium, and finally travel downstream to the ventricles. In this way, the heart contracts regularly, following a regular rhythm that is called sinus rhythm. In atrial fibrillation, the signals that start from the atria are unorganized and chaotic. Eventually, these signals are transmitted downstream to the ventricles. In atrial fibrillation, due to this chaotic conduction in the atria do not contract properly. 1 in 3 adults are at risk of developing atrial fibrillation during their life, and this risk increases in time. I'm sure that everybody would like to become just old and happy like this couple here, However, the more you grow old, the higher are the chances of developing atrial fibrillation. Most importantly, atrial fibrillation is a progressive disease. This means that if not treated, atrial fibrillation can become permanent. This is caused by a process called cardiac remodeling, which is characterized by abnormal changes in the electrical and structural properties of the heart. As Billy will explain to you later, these changes happen over time and contribute to make atrial fibrillation permanent. In this thesis, we pay particular attention to the structural changes that happen in the heart. Many studies have shown that these structural changes can impair the electrical activity of the heart and can support both the development or the stabilization of atrial fibrillation. Some people with atrial fibrillation do not have any symptoms. Others experience dizziness, shortness of breath, and even chest pain. Most importantly, atrial fibrillation is accompanied by a condition called hypercoagulability. This complicated term, hypercoagulability, refers to an increased tendency of the blood to coagulate. When the blood coagulates, it goes from a fluid form into a gel. Blood coagulation is essential to protect the body from excessive bleeding. And so in healthy individuals, there is a delicate balance between the activity of procoagulant factors and anticoagulant factors. In hypercoagulability, this balance is disrupted. In fact, the tendency of the blood to coagulate is abnormally increased. Hypercoagulability in atrial fibrillation increases the risk of blood clot formation. However, atrial fibrillation itself may also support this risk. In fact, in atrial fibrillation, the atria do not contract properly, so the blood pools in the atria. This mechanism, together with the hypercoagulability, further increase the risk of blood clot formation in the heart. In the worst scenario, if this blood clot leaves the heart and travels to the brain, it can cut the oxygen supply to the brain, leading to a stroke. So we know that atrial fibrillation is accompanied by hypercoagulability, and this increases the risk of stroke. In this thesis, we aim to demonstrate that hypercoagulability can also play a role on the onset and progression of atrial fibrillation. The blood contains important enzymes that are called coagulation factors. As the name suggests, the main activity of the coagulation factors is to help the blood to coagulate. In hypercoagulability, their activity is abnormally increased, so there is an increased risk of blood clot formation. The coagulation factors can interact with different organs of the body, including the heart. The heart is made of different cell types. One of these are cardiomyocytes. These cells are responsible for the contraction of the heart. Another important cell type are cardiac fibroblasts. These cells produce collagen, which is the scaffold of the heart, but they also control important processes such as inflammation. In this thesis, we were able to study cardiac fibroblasts by making use of cells isolated from the heart of laboratory animals and from heart biopsies of patients undergoing cardiac surgery. We were able to isolate and culture these cells in the laboratory and use them to study the effect of the coagulation factors. In this thesis, we found that coagulation factors can interact with cardiac fibroblasts and change their behavior. In fact, under the influence of the coagulation factors, cardiac fibroblasts can become activated, increase collagen, and increase inflammation. Together, these mechanisms may support structural changes in the heart. So with these observations, we demonstrated that coagulation factors can play a role on cardiac remodeling processes in atrial fibrillation, and so that the link between atrial fibrillation and hypercoagulability can be bidirectional. Next, we wanted to investigate if inhibition of the coagulation factors could also prevent cardiac remodeling processes in atrial fibrillation. We investigated this aspect in the goat model of AF. We used goats because their heart is very similar to the human heart, and so we included 3 groups of goats. In the first group, we included only healthy goats. In the second group, we included goats which were implanted with an electrical stimulator to induce and maintain atrial fibrillation for 4 months. In the third group, we included goats with atrial fibrillation, but which were treated with an anticoagulant, which blocks the coagulation factors. We looked closely to the heart of these animals, and we saw that in goats with 4 months of atrial fibrillation, cardiomyocytes appear bigger compared to the cardiomyocytes of healthy goats, suggesting the presence of ongoing cardiac remodeling processes during atrial fibrillation. However, when we look at goats with 4 months of atrial fibrillation treated with the anticoagulant, we saw that the size of the cardiomyocytes was comparable to the size of the cardiomyocytes of healthy goats, suggesting that inhibition of the coagulation factors via the anticoagulant can indeed prevent cardiac remodeling processes. In this thesis, we also wanted to understand the effect of atrial fibrillation on the coagulation system. Specifically, we wanted to see if atrial fibrillation was able to alter the goat coagulation balance. In the clinic, there are different ways to study the blood that is coagulation. Surprisingly, although these methods work for different experimental animals, they did not work for goats. So we had to find a way to study the goat coagulation system. The coagulation process involves a chain reaction that results in the activation of the coagulation factors and ends with the formation of a blood clot. It is important that this chain reaction is activated by a trigger. Without this trigger, the coagulation process cannot start. In this thesis, we found the appropriate trigger to start the coagulation process in goat blood. And so we developed a method to assess the goat coagulation potential. This means that we were able to measure the capacity of goat blood to coagulate when activated by the appropriate trigger. Using this method, we found that in young goats with 4 weeks of atrial fibrillation, there were no changes in their coagulation potential. However, we found that only the combination of advanced age with 4 weeks of atrial fibrillation led to an increase of the goat coagulation potential. Moreover, we found that only in all goats, 4 weeks of atrial fibrillation led to bigger cardiomyocytes and more collagen compared to all goats without atrial fibrillation. Together, these observations suggest that in elderly, the effect of atrial fibrillation on the coagulation system and structural changes may be enhanced. In summary, we know that atrial fibrillation is accompanied by hypercoagulability, and these will increase the risk of stroke. In this thesis, we found that the effect of atrial fibrillation on the coagulation system might be enhanced by aging. Moreover, we found that hypercoagulability can play a role on the onset and progression of atrial fibrillation. In fact, activated coagulation factors can interact with the cells of the heart and lead to structural changes which can further support atrial fibrillation. So if we go back to our initial question, which comes first, the chicken or the egg, people tend to say that there is no real answer to this question. In this case, if atrial fibrillation comes first, it will increase the risk of hypercoagulability, while if hypercoagulability comes first, it may accelerate the onset and progression of atrial fibrillation. So when it comes to the question which comes first, well, in this case, there is an answer, and it is that both atrial fibrillation and hypercoagulability can come first. Most importantly, one can promote the onset or progression of the other. With this, I would like to thank you very much for your attention and give the word back to Prorector.
Thank you very much for your clear presentation. We will now start with the opposition, and the opposition will be opened by Professor Reutelingsperger, Professor of Biochemistry of Apoptosis at this university.
Thank you, Mrs. Prorector. Dear candidate, first of all, I would like to start with congratulating you and giving you my compliments for this beautiful thesis and the excellent work, and also your presentation was very, very impressive. Thank you. The compliments go also to your promotion team. I would like to discuss with you some interesting, what I think is interesting, aspects of chapter 3, where you investigated the interaction of coagulation factors with cultured cells. And you looked at factor 10A, as you explained in your presentation, one of the enzymes of the coagulation system, and you looked at thrombin. Can you tell me, how did you pick the concentrations of these enzymes that you've used in this chapter? So for thrombin, it's 8 picomolar, and for 10A, it's 100 nanomolar.
Highly esteemed opponent, thank you very much for your kind words, and thanks a lot for your question. I think I need to clarify that it was not in the aim of this study to compare the effect of thrombin and the effect of 10A, as we are well aware that those are two different enzymes and they potentially activate the power receptors, even the same power receptor, leading to a different result in different downstream signaling. We picked those concentrations on the basis of previous experiments and on the basis of what are the most common concentrations used for in vitro studies. So we cannot claim that those are physiological.
Okay. So you didn't make a dose response of these factors and then see from that select the most optimal concentration for your effect?
That was done in the preliminary study.
Okay.
And we chose the range of concentration based on the previous literature and also the study that our group has done before.
Yeah, then I do have a question about the translation or the power of translation to the in vivo situation, which is one of the main goals of your research, of course. You want to that the results of your research will help to treat patients better. The 100 nanomolar 10A is a very high concentration, which is probably never achieved in vivo. In order for the coagulation system to get up and running, I think you need in the order of picomolar of 10A. So what do you think is the significance then of your finding that 10A in your in vitro system has an effect on the PAR or via the PAR on the expression of certain genes?
Thank you for your question. Well, when I started these experiments, and to be honest also now, I was not aware of the exact concentration that 10A can reach in places where thrombin coagulation may happen. I'm not just talking about systemic coagulation, but also local activation within the interstitial space. So it may be that 10A concentration might be reach even higher concentration than the one that we tested. But indeed can also be the other way around. I believe that for certain targets we see such a strong upregulation, and I can speculate on the fact that we could still achieve maybe not as such a strong upregulation, but still an upregulation that can be biologically relevant with lower concentration of factor 10A. I tend to think that it may be, so we get this very early response of, for example, MCP1 at 4 hours. So I do not believe that all the 10A present in the cell culture system is already gone or has already activated all the power receptors. So it might be that indeed, if we try the same experiments with a lower concentration, we might still get such an upregulation.
Okay, but these experiments should be done, of course.
Yes.
Yeah, so I think the importance of your findings is that there is a molecular link between the coagulation system and that is in circulation and your cellular system that regulates the function of an organ, in this case the heart. So that is very important. But whether such a link occurs in vivo, that's also a very important question in order to find pharmacological routes of intervention. one of the things, this interaction between the coagulation system and your cellular system, you see an effect of 8 picomolar of thrombin, and you see an effect of 100 nanomolar of 10A. And the thought that occurred to you is there a contamination in my factor 10A preparation? Is there a contamination of thrombin? That is what you discuss in the paper. And you say there is no contamination. The contamination would be in the order of 0.0 or 0.008%. So can you explain why you came to the conclusion there is no contamination?
Thank you for your question. Well, that can indeed be true. There might be residues of thrombin in the factor 10A, in the purified factor 10A reagent that we use. Well, we mainly base that observation on the fact that we do test the combination of factor 10A with rivaroxaban, which we know is a direct factor 10A, quite specific factor 10A anticoagulant. And then if you look at figure 4, we tend to see a quite substantial decrease of the upregulation of these genes because of the effect of rivaroxaban inhibition on 10A.
Thank you. I'm satisfied with your answers. Thank you very much.
Thank you.
Give the word back to the pro-rector.
Thank you very much, Professor Reutlingsberger. The next opponent is Professor Schroen. She's Professor of Experimental Cardiology, also at this university.
Thank you. Dear candidates, also my congratulations on this beautiful thesis, and I much enjoyed your talk. And I think it's great that you're standing here together with your paronyms to defend this thesis. I have a question, and I would like to ask one of your paronyms to read for me proposition 9.
9?
Yes.
So, proposition 9. Today, we have discovered that everyone is born with dozens of genetic glitches. There are no perfect human specimens, but not all our glitches are the same. So one treatment often does not fit all sufferers of a given disease.
Thank you so much. Now my question is, I fully agree with you. We are all born with our own genetic glitches. And indeed also more and more diseases are related or run into families. That's recognized. So also AF. What do you think about the contribution of genetic factors to AF susceptibility, independent of the presence of heart failure?
Of heart failure?
Yes.
Highly esteemed opponent, thanks a lot for your kind words and for your question. Well, the reason why I wanted to include this proposition is that the type of research that have done together with my colleagues focuses on very elementary, fundamental mechanisms by which atrial fibrillation is related to indeed cardiac remodeling. And this is very important and relevant. However, there is another side of the coin that is characterization of different substrates of atrial fibrillation. So in that case, genetic variants may affect these different substrates. So then characterization on a cellular level and also on a genetic level of atrial fibrillation substrate might be important as well. So in relation to heart failure, well, I think we also observe that atrial fibrillation and heart failure have, in a way, similar outcome on cardio remodeling processes, but they are triggered by different pathways. So genome-wide association studies may help to understand why, in the end, we see similar cardiac remodeling process, but they are triggered by different stimuli.
Yes, I fully agree with you that GWAS studies may shed light, but we have been trying this for many years already, and it seems to be quite a challenge to maybe ask the right question. Or what is your interpretation of the success rates of GWAS studies?
Well, I believe that, as you just mentioned, important knowledge has been gained by those studies, such as the genetic variants associated to PTX2. What I think that in combination with genome-wide association studies, we could improve in vitro cell culture on iPSCs, and maybe using CRISPR-Cas technologies to further dive into the biological meaning of that specific genetic variant. So that might help to maybe enhance or improve or expand even more the knowledge of the genome-wide association studies.
Yes, and I'm happy that you're mentioning iPSCs already, because that was my next question. So how do you see that, the use of iPSCs for AF research? Do you intend to focus on certain aspects of the use of iPSC?
Thank you very much for your question again. I think it's really important to focus on that as well. So in our thesis, we use two different cellular models of cardiac fibroblast, rat adult cardiac fibroblast and human cardiac fibroblast, both primary cells. However, although well characterized, they still showed many limitations. And for example, the human cardiac fibroblast, well, one of the first limiting factors was the limited amount of donors. Another possible limiting factor is that we don't really have a healthy control for those cells. So in that sense, the iPSCs, I think, will contribute extensively to improve these methods.
Yes, but then you still have the issue of healthy control, because who is healthy?
Yes.
How do you define that? Yeah.
Yeah, I believe that we need a deeper characterization of what is healthy and what is not, and maybe it can be a very broad question. Maybe we can restrict to that question to specific comorbidities might interfere with the
a philosophical question, and if you would have all the money in the world, very smart question, of course. What would you do to solve the AF puzzle in combination with genetic issues?
Sorry.
What would you do if you have all the money in the world to solve the AF puzzle, like the substrate issue, the genetic contributions? Where would you start?
Well,
just very brief.
Thank you very much for your question. If I had all the money in the world and a limited amount of time as well, I would focus on the most advanced techniques, such as single RNA sequencing, single cell RNA sequencing, for example. I think it's interesting to see how the different subpopulation of cardiac fibroblasts and other type of cells of the heart with different degrees of atrial fibrillation and different substrates. And expand, of course, some important observations of this thesis, which are more hypothesis generating as well.
Well, I would agree, as long as you take along the genetics. I'm happy with your answers, and I give the word back to the prorector.
Thank you very much, Professor Schroen. The opposition will be continued by Professor Goethe, Professor of Cardiology at St. Vincent Hospital in Paderborn in Germany, and Professor Goethe is present online. Welcome. The floor is yours.
Yes, I'm honored, of course, to be in Maastricht, at least online. Sorry for not being there. So I really enjoyed, dear candidate, your presentation. And I have two questions which are a bit translationally related to the clinical aspect. So you showed quite nicely in the first chapters of the thesis also that the PAR receptor is one target really to have an inside out or outside in signaling from the clotting system into the myocytes or the fibroblasts. So my question is, you blocked in those approaches the clotting factor per se, but how would the experiments look like if you would block a PAR receptor by the developed PAR receptor antagonists, which were in the first development of clinical use, like Vorapaxar or so?
Hi, esteemed opponent. Thanks a lot for your kind words and for your question. Indeed, this is a very important aspect to take into account. We inhibited a coagulation factor, but we might think about an alternative strategy and inhibit the PAR receptor. Well, I think both are valid strategies. However, if we think about, if we translate these to clinical situation, I'm aware that, for example, Vorapaxar has also an anticoagulant effect and also has a bleeding risk, as well also rivaroxaban. So, first of all, if I have to choose between PAR1 antagonist and rivaroxaban to prevent cardiac remodeling and PARS activation, I would always consider, first of all, the risk of stroke and what is known by clinical studies on the efficacy of the drug and on the bleeding risk profile. So I would, for example, go for rivaroxaban. On the other hand, I also know that rivaroxaban blocking factor 10A might not only prevent activation of PAR1, but also other PAR receptors. So that may be something to take into account. And the other way around, Vorapaxar inhibiting only PAR1 might not be sufficient to prevent some remodeling processes.
Thank you. But with regard to the effect of rivaroxaban, for example, on the cellular aspects, do you think that it's really necessary to have the full anticoagulant activity of rivaroxaban, or would you suggest or assume that the tissue effects occur at way lower concentrations?
Very much for your question. This is indeed very interesting, and it's also one of the aspects that I wondered during my PhD. I would definitely test whether a lower dosage of rivaroxaban, for example, also in the GO model, that would prevent cardiac remodeling process without indeed increases the bleeding risk and without affecting the coagulation. So that's a very interesting and important strategy that can be taken and should be taken, first of all, in preclinical studies indeed.
Yeah, thank you. So, and my last question is, I mean, you explained it very nicely, the egg and chicken problem. So just probably to have one more idea would be, do you have information, or what would your answer be in patients with a genetically determined clotting factor abnormality triggering deep vein thrombosis or so? So patients who have constantly increased risk to suffer a thrombotic event. Would you expect that these patients also have a high incidence of atrial fibrillation during their lifetime? Or a higher incidence?
Thank you. Thanks a lot for your question. This is a very indeed important consideration. I believe if you have a genetic modification that predisposes to thrombosis, you might have upregulated processes such as inflammatory processes in the heart and in the systemic circulation that indeed could eventually lead to also cardiac remodeling. So needs to be, of course, investigated, and I'm just speculating on these, but that might happen indeed.
Okay. So thank you so much for your answers. I'm satisfied with this.
Yeah.
Thank you very much, Professor Goethe. The opposition will be continued by Dr. Van Eijs. He's Associate Professor Genetics and Cell Biology at this university.
Congratulations with the finishing of your PhD study. I am struck by the cover. It's a very nice cover, a very Italian goat. We don't have them like this in the Netherlands. They're more black and white over here. So it was a pleasure to read the thesis, but I have stumbled into a number of things. And one of the questions I ask myself is, you taking samples for gene analysis, for expression analysis, and I have been wondering, in those samples you take from atria, there are more types of cells. How do you differentiate between what is happening in the different cell types?
Esteemed opponent, thanks a lot for your kind words and for your question. Indeed, for the studies in the gold model of atrial fibrillation, we take entire atrial tissue and we perform RTQ-PCR on that. From those specific results, we cannot speculate on what is the contribution of specific cell types. And actually, this is a very interesting aspect, and as I was just mentioning before, one way to differentiate would be single cell RNA sequencing, for example. We can only speculate on that on the basis of what we see, for example, in chapter 3, when we look at two different species and we look at cardiac fibroblasts from rats and human cardiac fibroblasts, and we see similar expression of PAR receptors, for example. So I might dare to say that if we look at goat cardiac fibroblasts, these may be similar, but again, I think single-cell RNA sequencing is much better and more sophisticated technique to respond to that question.
Yeah, the work has been done by RT-PCR, and I have been wondering why there is no follow-up, let's say, with immunomarkers, things like that, on the protein level.
Thanks a lot for your question. This is something that we planned and we are still planning to do, validating what we see at the gene expression level with the protein analysis. At that specific moment in time, due to limited amount of time as well, we couldn't really invest more time and effort in developing anti-GOATs antibodies that could be really reliable in detecting those markers in vitro. But still, we wanted to have at least a general overview of the pathways that are. So this is something that we are planning to do, and I will help to do as well.
And then comes my main question. On page 14, you present a model, a hypothesis on what's going on in the fibrillating heart. And then on page 158, there is a more complicated model, and you're sticking out your head there, because most of that model is the same as the old model, except lines that are dotted, or the only firm line is lone AF. Yeah, and the thesis goes about hypercoagulability and the, let's say, the influence of that to atrial fibrillation. But where is that arrow? Where do I find, for instance, an arrow directly going from the clotting back to atrial fibrillation? Does that always have to go via the cardiomyocytes and the fibroblast? Or do we have a direct effect for which you have been, let's say, mentioning a number of times during these different chapters?
Thank you for your question. Well, when we draw this scheme, of course, we had in mind a loop, a circle. And indeed, as you observed, there is a connection between hypercoagulability through PARS activation. So it might be not the best representative scheme, but the direct link from hypercoagulability to AF is going via PARS activation. So that is the direct link, and what we try to investigate there, and what we hope to explain in our thesis, together with the presence of comorbidities that can, of course, explain even more the bidirectional link. Does that answer your question?
No. You see, the old scheme is basically the, if I restrict myself to the upper left part, Yeah, that is the work that has been done 25 years ago by Osma and Wijfels and that group. Yes? And so now the question is, what do we add by, or let's say, what is the reason behind taking up things like clotting and factors like factor 10 in this scheme? Because I do not find very much back of that in the new scheme.
Well, maybe then it's better if I explain it with my own words. What I believe is the novelty. So you cited very important and fundamental data. What the novelty in this study is that we link an alternative pathways that can contribute to remodeling through the coagulation factors and the PAR receptors. So in this way, our studies indeed provides many observations that are mainly hypothesis generating. So I think that in that case, answering or diving in more into those observations may be helpful to clarify even more this thesis and the observation taken from this thesis. What it's novel is that for the first time in such large animal model like the goat model of atrial fibrillation, we do see that systemic inhibition of a coagulation factors can prevent one of the most known and studied cardiac remodeling process, that is hypertrophy. So that, I believe, adds up to the very important knowledge already gained in the years before.
Okay, but you still have some dotted lines to fill in, right?
Of course.
Some work to do. Thank you.
Thank you, Dr. Van Eijs. The opposition will be continued by Professor Wenzel. He's Professor of Vascular Inflammation, Department of Cardiology of the University of Mainz in Germany.
Thank you, dear Pro-Rector. Dear candidate, thank you for your presentation. I was very much pleased by your presentation. I have a couple of questions. So in your thesis, you focused on factor 10A and factor 10A inhibition. My first question is, can you maybe discuss with me whether factor 2 inhibitors, which are also in clinical use to prevent atrial fibrillation-associated stroke, have comparable effects on your GOAT model?
Highly esteemed opponent, thanks a lot for your kind words and for your question. Yeah, so in our study, indeed, we use an anticoagulant that directly blocks factor 10A, and this, as a consequence, would also prevent thrombin formation. We did not investigate in this specific study what thrombin would do, but looking at the experiments that we perform in chapter 3, we can only speculate that there are some pathways and some processes in which thrombin and 10A might be not equivalent but very similar. However, from literature, we know that even the activation of the same PAR receptors by different enzymes may lead to a different outcome. So it is very difficult to speculate right now about this. I would say that it's interesting to really differentiate what are the processes that might be activated by the same coagulation factors and by this activation of the same receptor, and what they could be different. In fact, we see that thrombin and 10A, for example, upregulate two important pro-inflammatory mediators like MCP1 and IL-6. So in this sense, I can speculate that maybe on an inflammatory process, maybe the two enzymes might have similar, to some extent, effects. But of course, this also needs to be further clarified.
Yes, this question also relates to the fact that you use the CUT assay to, if I'm correct, right? in your work, and this also tests thrombin propagation or thrombin potential. So that is why I was asking this question. Maybe this is something for future studies. My second question goes to similar direction. So there are novel drugs that target factor 11, other protein that is a coagulation protease, which has been shown to promote thrombin amplification in vivo and also in vitro. And could you speculate whether factor 11 inhibitors that are now in clinical trials already have a potential benefit in your model on atrial remodeling and maybe also on PAR activation and AF progression?
Thanks a lot for your question. This is also an important observation to make indeed. Well, at the best of my knowledge, I'm not aware of mechanisms that link activation of PAR receptor with factor 11. Nevertheless, targeting factor 10A might indirectly also inhibit the positive feedback loop on factor 11. So we might still be able to prevent any possible potential cardiac remodeling processes triggered by 11 via inhibiting 10A on thrombin. So yes, at the moment I don't have data about that, but for sure it's an interesting aspect to consider, and maybe further investigation of the activation of PAR receptor by other coagulation factors that are not known to activate PARs can be an important first step.
Yes, because this also implies the importance of factor 2, like of the thrombin, right? So that is maybe one of the central proteases here in atrial fibrillation remodeling, maybe. In addition to factor 10A. And then my next or last question maybe refers to proposition number 3, and I would ask one paranymph to read the proposition. Number 3.
Yeah, proposition number 3. Systemic factor 10A inhibition during AF locally protects the heart from atrial structural changes that play a crucial role in the pathogenesis of AF.
So thank you very much. So my question is, did you study or do you have an idea about what are the short or long-term effects of AFib and of factor 10A and or 10A inhibition on structural and functional changes of the ventricular myocardium, not only the atrial myocardium?
Thank you very much for your question. Well, it is indeed clear that we did not investigate any structural changes of the ventricles, mainly because of our hypothesis, but we do not exclude further investigation of ventricles as well. What I do know, at least from preclinical studies, is that rivaroxaban was able to prevent left ventricular hypertrophy in pressure overload mice model. So there can be, of course, a protective effect of anticoagulation also on ventricles, and this leads to the observation that indeed the activity of the coagulation factors and other proteases that activate PAR receptors might not be specific for atria, but also for ventricles.
Thank you very much. I am happy, very satisfied with your answers, dear Pro-Rector. Thank you.
Thank you, Professor Wenzel. The opposition will be continued by Dr. Nagy. She's a postdoc at the Department of Biochemistry at this university.
Thank you, Ms. Proctor. First of all, I would like to congratulate to your candidate. It's very impressive work, and yeah, the cover is, I think that's one of the happiest cover I ever seen in a thesis. I also would like to congratulate to your supervisory team. But before I start my question, I would like to ask one of your paranames to read proposition 4.
Okay, proposition 4. Assessment of coagulation activity in peripheral blood is insufficient to estimate the impact of procoagulant effects locally in the atria or in the interstitial space of the atrial myocardium.
So in this proposition, you basically refer to that you really have to measure everything locally, but have you actually measured, how would you measure locally in a patient situation, for instance, the present hypercoagulability?
Esteemed opponent, thank you very much for your kind words and for your question. Actually, some people and a research group have tried to measure differences in the activation of the coagulation potential between the atria and versus to the systemic circulation. That has been done in patients and has been done in paroxysmal atrial fibrillation undergoing ablation. So the striking result from that specific study is that during ablation, a collection of blood sample from the left atrium resulted in increased thrombin-antithrombin levels and platelets activation compared to blood sample taken from the systemic circulation at the same time point. So this is also a justification why I wanted to include this proposition in my list of the propositions, because we assume that the activation of the coagulation happens, it's uniform through the body, and it might not be the case in atrial fibrillation, for example.
Yes, but you refer to this proposition as it's coming from your thesis. So actually, how did you evaluate the systematic coagulation in your case? So what did you use to measure it?
We took blood from the jugular vein of goats, and then we used that to make poor platelet plasma, and so we used the thrombin generation to look at the potential, so an indication of increased potential for coagulation.
But then do you talk about potential or activity? Because you also state in your thesis multiple places that the thrombin generation assay actually only measures potential and not actually reflecting in vivo activity.
Yes, thank you very much for your question. And this is a very important consideration to make. Thrombin generation gives an overview of the general coagulation potential indeed, so the capacity of blood to coagulate. And it does not really say what exactly is happening in that specific moment in time on a level of activation of the coagulation. So this is something that we hope to do, and we hope to do in the past, and we still hope to do, developing assays that could look at activation of the coagulation, and that together with thrombin generation might complete the overview on the effect of atrial fibrillation on the coagulation system. So Elisa assays for activated coagulation factors and modifications of the thrombin generation.
And also because, I mean, in stroke, that's a bit of a similar situation, of course, because there's always a discussion whether you can pick up systematically also hypercoagulability. And there are also some studies showing that actually you can, and there, due to the blood-brain barrier, of course, it's not, you not necessarily expect this, or at least there are some insufficient knowledge about it, how precise it is. So then do you think that if you could sensitize your assays, then you would be able to also pick up systematic effect easier?
Through the thrombin generation?
To any assays what you would like to develop, for instance?
I do believe that improvement of the assays would definitely pick up local changes, but I also want to add that when I refer to local changes, I also refer about activation of coagulation that may happen within the tissue. So in that sense, we can still use a thrombin generation, but maybe other techniques might be more sensitive.
Thank you. I'm happy with the answer, and I would like to give back the word to Proctor.
Thank you, Dr. Nagy. The opposition will be continued by Dr. Blankenstein. He's Associate Professor of Pharmacology at this university.
Thank you very much, Pro-Rector. Yeah, I also would like to compliment you on a very nice thesis. Not only the cover, but also the content, I think, is very nice. And I would like to extend my congratulations to your promotion team. Yeah, for my question, I would like to focus on chapter 5, where you use a treatment with rivaroxaban to interfere with the AF development in your goat model. And actually, in chapter 5, but also in your introduction, you clearly stated that normally persistent AF and structural changes of the atrium go hand in hand. But if I look in figure 3 and 4 on page 93, there is actually not much fibrosis going on at all in your goat model. And your myocyte size increases, so there is hypertrophy in the AF group, and in your rivaroxaban group you can reverse that to normal, but that doesn't mean that your AF goes away, because you still see signs of AF in your electrophysiological evaluations. So yeah, my interpretation of this is that there's kind of a dissociation here between the structural changes that you described and the electrophysiological problem of AF. And I would like you to comment on that.
Elissimo opponent, thanks a lot for your question and for your kind words and compliments. Yes, there are different ways to address your question indeed. It's a very complex question as well. What we observe in the goat model of 4 months of atrial fibrillation is indeed a more pronounced cardiomyocyte hypertrophy, while we seem to not pick up any changes in fibrosis. Well, we observed that there are changes of profibrotic processes in the atria. We picked them up with RT-qPCR, looking at the upregulation of collagen and alpha-smooth muscle actin, which can indicate that it might be that is in this moment in time we are too early to see macroscopically fibrosis. Mm-hmm. The other question point that you touched on is why don't we see less complex atrial fibrillation in response to the rivaroxaban treatment? Well, also here I can only speculate on possible explanations. What I do believe is that it also may be that, again, we are too early in time, and maybe this 1 micrometer difference in diameter is not yet sufficient to trigger or increase complexity of AF, which can be reversed by rivaroxaban. So it would be interesting to look at a longer model of atrial fibrillation, or even implement the rivaroxaban anticoagulation in the aging study.
Yeah, thank you. That's actually a very interesting point you raised, because that would have been my next question. In the chapter that follows this one, you use a model of aged goats, and there actually you do see the fibrosis and also the hypertrophy. The only problem is that you didn't use the rivaroxaban there. So my question would be, what would you expect if you would test your rivaroxaban in a more aged model of atrial fibrillation?
Thanks a lot for your question, and this is very interesting because it's also one of the plans that we hope to do. Mm-hmm. So what we see in the aging goat, that this increase in myocyte size is bigger if you compare to what you observe in the chapter 3, but also is accompanied by an increased coagulation potential. So what I can only speculate on is that with an anti-10A inhibitor such as rivaroxaban, we might still be able to see a decrease in atrial cardiomyocyte size. But again, there are many open questions there. The fact that we have older animals maybe lead to a different outcome. For sure, it's something that we want to further investigate.
Yeah, and regarding the drug that you've chosen, that refers a little bit to the question that Professor Wenzel also asked. You choose for a very specific factor 10A inhibitor. In the past, compounds like acinocomerol were used in this context, and they have a much broader inhibitory effect on different coagulation factors. Would that not be a choice that would lead to more success in this context?
Thank you very much for your question. Indeed, those are also very valuable anticoagulants. However, there are indications that Undercarboxylated coagulation factors may still be able to trigger PAR receptors activation in a non-membrane dependent way. So if that is the case, in fact, with a direct inhibitor such as rivaroxaban, we can target not only activation of the coagulation, but also remodeling through PAR activation. That's why we decided to investigate that.
Okay. Well, thank you very much. I'll give the word back to the prorector.
Elisa D'Alessandro, the time appointed for defending your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and your defense. Of course, because we have another thesis defense, it will take a little bit longer before you hear the outcome of our deliberations. But I request you and your company to await the results of our deliberations and our return. Thank you.
Please, all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes. In the meantime, let's take a closer look at Maastricht University's renowned teaching method, problem-based learning. Once the prorector reconvenes the session, we'll tune in and continue the live stream.
Problem-based learning, or PBL. What does that mean exactly? 3 of my fellow students and I will show you around. Every week, we analyze a different case or issue together. We discuss the case and everyone can contribute different perspectives to the group discussion. If we get stuck, our tutor helps us out and suggests what we could do next. I prefer going to the library to prepare. Here, I can focus and I have quick access to books or journals that help me understand the case. Today, I can also train my stitching skills in the Skills Lab, where you can immediately put into practice what you've learned. After a day like this, I like going to the gym to clear my head and get ready for the next day.
At UM, you meet people from all around the world. Hello guys. Some of them are doing their exchange semester here, and they often say that PBL helps them learn and retain things very easily. I can understand why. It's a very active way of learning because you have to bring your own perspective to real-life cases. You have a lot of freedom to manage your time, your studies, your hobbies, and your work. Of course, that also means a lot of responsibility. Right now, for example, I'm arranging my exchange semester in Madrid. How cool is that? In this group session, we're the managers who have to allocate the resources of a real company. This is how we put into practice what we learned this morning. Studying here means being proactive and learning to plan well. Prioritizing and performing well under stress are great skills that help you develop as a person. But now, it's time to grab a coffee at the Student Service Center.
Right now, we're at the Brightlands Camelot campus. Here we can apply knowledge from lectures and tutorials in a practical setting. This helps us understand what we have learned and further develop our lab skills. Today, we're determining the amounts of cholesterol in various products. What I really like is the project periods at the end of each semester, where we complete a full research project. That includes planning, collecting data, analyzing, and presenting the findings. That way we learn how research works and we're able to see what it's like to be a real scientist. After practicals, I have to write a lab report that also helps me process everything I have learned today. UM has a lot of learning spaces where you can work on your own or with other students. This evening, I'm meeting my friends for a movie night organised by the MSP Study Association.
If you study law, you have to read quite a lot. Not all information is relevant, so you learn how to easily find the information you need to solve your case. In the afternoon, I have to give a presentation, so I like to practice it with a fellow student. Later today, a lawyer is giving a lecture. This will help us better understand the case we're working on. Speaking in front of a group is quite exciting the first time round, but you get used to it quickly. And having to present helps you also to adopt knowledge better. What I like the most is that we sometimes get to enter a plea in front of a real judge. These moot courts are really exciting. Now it's time for drinks with friends. Studying is important, but so is relaxing once in a while.
Who are we as a UM community? We are a group of people who love,
talk,
move,
learn, work and think in a variety of ways.
And we all want to be a valuable and valued member of our UM community.
a community where everyone can feel safe, needs nurturing.
To bring a community to its full potential, we all have a role to play.
Every single one of us needs to be part of a moving conversation.
To understand each other.
To find help if you need it. Or to give help when asked.
To connect with your peers and share some stories.
And have some fun at the same time. Make space for new perspectives and broaden your horizon.
Discuss. We don't have to agree, but we can learn.
That diverse views are at the basis of our understanding.
And including these views are the basics of a healthy community.
Make a difference at your own pace.
Join one of our active groups or become an ally. The first steps are not always easy, but let's help each other on this journey.
Do you want to know more? Follow the UM's Diversity and Inclusivity Office on social media.
Or check out maastrichtuniversity.nl/diversity.
Hi, today we'll talk about sex and gender. Two words which are often used interchangeably in research, but however have separate meanings.
In this video, we'll talk about the difference between sex and gender, why it is important to account for one or the other in research, and how to do it.
Okay, let's start. What is sex and what is gender? Sex is assigned at birth and has mostly to do with physical and physiological features such as having XY chromosomes or having a uterus. While gender relates to your experienced or constructed identity, the way you feel about yourself. It is important to know that for some people the gender identity might not always align with their sex assigned at birth. Concerning sex, there are three options: male, female and intersex. The most common genders are man and woman. A majority identifies as either one of those. But there are many other genders next to man and woman, and sometimes people identify as both, or as neither man nor woman, as they might identify with another gender. Furthermore, people's gender identity can be fluid. This means that the way they identify themselves can change over time. Gender identity can be very personal, and everyone can have their own interpretation of their gender identity.
But why do we need this distinction in research? Sometimes in a study, you want to compare the results of two groups and see if there are any differences that are relevant to know. It is important to ask yourself the question, do you want to know the biological differences or some behavioral differences? For some studies, it is relevant to know the biological differences. For example, symptoms of heart attack can be different for males and females. And for some studies, it is relevant to know the behavioral differences. For example, do the same kind of advertisements work well for people identifying as men or women?
So, how can we more accurately account for this in research? We would like to give you some hands-on tips for how to use the words sex and gender right and to make your research more inclusive. First, if you want to address people in general, you could be more inclusive and use participant or reader. In Dutch, beste deelnemer, lezer.
Second, ask yourself, is it necessary to obtain this type of information? What do I need it for? And how will I use it? Not every study benefits from collecting data on sex or gender, so then you should not include these questions.
Third, if for your study it is relevant to know more about the biological sex, you might ask, which sex was assigned to you at birth? Or in Dutch, welk geslacht is jou toegekend bij je geboorte? Don't forget to include the option of intersex.
Fourth, if you're more interested in behavior, you can ask the open question, what label do you feel best describes your gender? Or in Dutch, welk label beschrijft jouw genderidentiteit het beste? And leave the answer options open.
But note, when asking for someone's identity in closed format, you should always add the answer options not listed namely and I'd rather not say, or in Dutch, niet genoemd namelijk en zeg ik liever niet. It can be a sensitive question for people, since discovering your identity is not always a straightforward process.
We hope to have given you some new information and tips on how to make research more inclusive. This video was brought to you by UM Pride. Feel free to reach out if you have any questions.
We now continue this academic session, in which now Billy Scaf will defend the academic thesis, Atrial Fibrillation and Hypercoagulability, a two-way street with many side roads. May I invite you to present a summary of your studies and the conclusions of the thesis.
Dank u wel, mevrouw de prorector. Geachte leden van de corona, geachte familie, vrienden en collega's, bedankt voor jullie aanwezigheid vandaag hier in deze mooie aula van de Universiteit Maastricht aan de Minderbroedersberg. Maar ook bedankt voor degenen die online meevolgen. Het komende kwartier ga ik jullie een samenvatting geven van het onderzoek zoals beschreven in mijn of ons proefschrift getiteld Atrial Fibrillation and Hypercoagulability, a two-way street with many side roads. Het onderzoek zoals beschreven in ons proefschrift is het resultaat van een unieke samenwerking tussen Elisa en mijzelf, maar vooral ook tussen onze afdelingen, biochemie en fysiologie. En voordat ik de belangrijkste bevindingen met jullie ga delen, geef ik jullie een korte introductie over de ziekte atrium fibrilleren. Wat is atriumfibrilleren? Atriumfibrilleren wordt afgekort met AF en is onder velen van jullie wellicht beter bekend onder de term boezemfibrilleren. Deze ziekte dankt zijn naam aan de locatie in het hart waar het zich afspeelt. Hier zien jullie een schematische weergave van een doorsnede van het hart. Dat bestaat uit 4 ruimtes. De bovenste 2 ruimtes zijn de atria, oftewel de boezems, en de onderste 2 ruimtes zijn de ventrikels, oftewel de kamers. Zoals jullie wellicht weten, wordt het hart aangestuurd door elektrische signalen. En deze elektrische signalen leiden tot spierbewegingen waarmee het hart het bloed door het lichaam kan pompen. In een gezonde situatie vertrekken deze elektrische signalen vanuit de sinusknoop. Dat is de grote gele stip in het rechteratrium. Vervolgens verspreiden die signalen zich in de richting van het linkeratrium en in de richting van de AV-knoop. Dat is de kleine gele stip in het midden. Vanuit de AV-knoop verspreiden deze signalen zich in de richting van de ventrikels. En deze vaste volgorde van elektrische activaties resulteert in een regelmatige hartslag, ook wel sinusritme genoemd. In de situatie van atriumfibrilleren is de elektrische geleiding in de atria verstoord. Dit resulteert in onregelmatige samentrekkingen, ook wel fibrillatie genoemd, en dat vermindert de algehele pompfunctie van het hart. Op deze bewegende beelden is nog beter te zien hoe iedere atriale activatie wordt gevolgd door ventriculaire activatie, en deze vaste volgorde van activaties resulteert dus in de regelmatige hartslag. In de situatie van atriumfibrilleren zijn het de snelle en onregelmatige activaties van de atria die ervoor zorgen dat de ventrikels zich niet goed kunnen vullen met bloed, waardoor er dus minder bloed naar de rest van het lichaam gepompt kan worden. Waarschijnlijk kent ieder van jullie wel iemand met atriumfibrilleren. Gemiddeld komt deze ziekte namelijk bij 1 op de 30 personen voor. En omdat we tegenwoordig steeds ouder worden, is de verwachting dat atriumfibrilleren het voorkomen van atriumfibrilleren de komende jaren alleen maar zal toenemen. Hoge leeftijd is namelijk de belangrijkste AF-risicofactor en dat wil zeggen dat AF vaker voorkomt bij oudere mensen. Andere AF-risicofactoren zijn onder andere hartfalen, diabetes, hoge bloeddruk, maar ook intensieve duursporten. Er zijn AF-patiënten die niet of nauwelijks symptomen ervaren, maar de meeste AF-patiënten beschrijven klachten als hartbonken, duizeligheid, vermoeidheid of een beklemmend gevoel op de borst. Het grootste gevaar van AF schuilt in het feit dat mensen met AF een vervijfvoudigde kans hebben op het vormen van een beroerte, oftewel een herseninfarct. En een herseninfarct ontstaat doordat een bloedstolsel vanuit het hart via de bloedvaten naar de hersenen gaat en daar de bloedtoevoer blokkeert. Atriumfibrilleren is ook een progressieve ziekte. Dat wil zeggen dat de ziekte langzaam erger wordt. AF-patiënten wisselen periodes van sinusritme en atriumfibrilleren met elkaar af en na verloop van tijd worden de periodes in AF steeds langer. Oftewel, periodes van sinusritme komen steeds minder voor. Het verergeren van atriumfibrilleren wordt gekenmerkt door verschillende veranderingsprocessen. In het begin zijn dit elektrische veranderingen, maar na verloop van tijd treden er structurele weefselveranderingen plaats. En deze weefselveranderingen die zijn met name belangrijk voor het verstoren van de elektrische geleiding in de atria en het stabieler of hardnekkiger worden van atriumfibrilleren. De weefselveranderingen die plaatsvinden tijdens AF gebeuren voornamelijk in het atriale spierweefsel. En als we daarop inzoomen, ziet dat als volgt uit. In het geval dat we een dwarsdoorsnede zouden maken van deze spierbundels, zien we cirkelvormige hartspiercellen. En het verschil tussen gezond en ongezond hartweefsel zit in het verschil tussen in het tussenliggende bindweefsel, ook wel fibrose genoemd, maar ook in het formaat van de atriale spiercellen. Het grijze weergave aan de rechteronderzijde is een schematische weergave van het type fibrose dat wij hebben onderzocht. En met name fibrose, oftewel bindweefselvorming, en het formaat van deze atriale spiercellen, die zijn belangrijk voor het verstoren van de elektrische geleiding in de atria. Nu dan, wat hebben wij onderzocht? Zoals ik zojuist aangaf, is er tijdens AF een vergrote kans op het vormen van bloedstolsels. Echter, in de gezonde situatie zou er geen aanleiding moeten zijn voor het vormen van bloedstolsels in het hart. Maar tijdens AF is die aanleiding er wel. En dat komt door een samenloop van verschillende omstandigheden. Een van deze omstandigheden is hypercoagulabiliteit, oftewel een verhoogde stollingsneiging. En een verhoogde stollingsneiging wordt gekenmerkt door een scheve verhouding in de aanwezigheid van stollingsenzymen in het bloed. En met name de laatste jaren komen er vanuit de wetenschappelijke wereld steeds meer suggesties dat deze stollingsenzymen ook betrokken zijn bij structurele weefselveranderingen. Dat zou dus kunnen betekenen dat deze stollingsenzymen ook kunnen bijdragen aan de progressie van atriumfibrilleren. Oftewel, atriumfibrilleren leidt niet alleen tot een verhoogde stollingsneiging, maar stollingsneiging zou ook kunnen bijdragen aan het verergeren van atriumfibrilleren. Wat hebben wij ontdekt? In hoofdstuk 3 beschrijven wij het onderzoek dat we hebben gedaan met geïsoleerde cellen uit het hart, de zogenaamde fibroblasten. Deze fibroblasten hebben we gestimuleerd met stollingsenzymen en vervolgens vergeleken met cellen die we niet hebben gestimuleerd met stollingsenzymen. En middels een genetische analyse, de inmiddels alom bekende term PCR-analyse, zagen wij dat de fibroblasten die werden gestimuleerd met stollingsenzymen reacties gaven die kunnen leiden tot bindweefselvorming en inflammatie, oftewel een ander woord voor ontsteking. Dat lijkt dus aan te geven dat cellen die worden gestimuleerd door stollingsenzymen kunnen bijdragen aan processen die bijdragen aan het verergeren van atriumfibrilleren. Naast het onderzoek met geïsoleerde cellen uit het hart hebben wij ook onderzoek gedaan met levende geiten. En wij hebben voor het diermodel van atriumfibrilleren gekozen omdat al sinds de jaren 90 blijkt dat het AF-geitenmodel representatief is voor het verloop van AF in mensen. In deze studie hebben we 3 verschillende groepen met elkaar vergeleken. 2 groepen waarin we voor 4 maanden atriumfibrilleren hebben opgewekt en 1 groep waarin we geen AF hebben opgewekt, oftewel die bleven in sinusritme. 1 van de 2 AF-groepen hebben we tijdens de periode van AF behandeld met antistolling. En dit type antistolling, dat trouwens ook bij AF-patiënten wordt gebruikt voor het verlagen van de stollingsneiging, werkt zeer specifiek op 1 stollingsenzym. Het doel van deze studie was om te kijken van of een verlaging van de stollingsneiging door middel van de antistolling zou het ontstaan van de weefselveranderingen kan vertragen en daardoor ook de progressie van atriumfibrilleren kan verminderen. Nou, wat hebben wij ontdekt? In hoofdstuk 5 beschrijven wij dus dat onderzoek wat we hebben gedaan na een periode van 4 maanden AF. En wij zagen dat de geiten met 4 maanden AF vergrote atriale spiercellen hadden in de atria. Wat wij ook zagen is dat de behandeling met antistolling het vergroten van deze spiercellen voorkwam. Interessant genoeg, en in tegenstelling tot wat we verwacht hadden, zagen we dat de stollingsneiging in de geiten met atriumfibrilleren niet verhoogd was. En een mogelijke verklaring hiervoor is dat de aanwezigheid van enkel atriumfibrilleren, dat is atriumfibrilleren zonder andere risicofactoren, toch niet voldoende is voor het verhogen van de stollingsneiging. Want in werkelijke AF-patiënten zijn er vaak meerdere factoren aanwezig die kunnen bijdragen aan het verhogen van deze stollingsneiging. Met dit in ons achterhoofd hebben wij het AF-geitenmodel uitgebreid met 2 belangrijke AF-risicofactoren, namelijk hoge leeftijd, oftewel ouderdom, en hartfalen. Opnieuw waren we benieuwd wat de aanwezigheid van deze AF-risicofactoren alleen of in combinatie met AF zouden geven op de stollingsneiging. En bovendien waren we benieuwd naar wat het effect was van de aanwezigheid van deze risicofactoren in combinatie of niet in combinatie met AF op de structurele weefselveranderingen en het daaruit volgende effect op de progressie van AF. In hoofdstuk 6 beschrijven wij het onderzoek dat we hebben gedaan naar de AF-risicofactor hoge leeftijd. En opnieuw hebben we dus gebruik gemaakt van het AF-geitenmodel, maar in tegenstelling tot het onderzoek zoals beschreven in hoofdstuk 5, hebben we nu gekozen voor een periode van 4 weken van atriumfibrilleren. En 4 verschillende groepen hebben we daarin vergeleken. Dat zijn jonge geiten zonder AF, dus in sinusritme, jonge geiten met AF, oude geiten zonder AF en oude geiten met AF. Wij zagen dat enkel de combinatie tussen hoge leeftijd en AF resulteerde in een verhoogde stollingsneiging. En bovendien ging dit gepaard met een vergroting van atriale spiercellen dat al plaatsvond na 4 weken. Dit lijkt dus aan te geven dat de combinatie tussen hoge leeftijd en AF de stollingsneiging versterkt en daarmee de ontwikkelingen op weefselniveau versnelt. In hoofdstuk 7, het laatste experimentele hoofdstuk van ons proefschrift, beschrijven wij het onderzoek dat we hebben gedaan naar hartfalen. Hartfalen wordt gekenmerkt door een verminderde pompfunctie van de ventrikels van het hart. Opnieuw hebben we gebruik gemaakt van het AF-geitenmodel en we hebben geiten vergeleken die enkel hartfalen hadden, enkel AF, en geiten die hartfalen hadden en waarin we 1 week AF hebben geïnduceerd. Opnieuw waren we verrast door te zien dat nog hartfalen met AF resulteerde in een verhoogde stollingsneiging. Maar nog bijzonderder was dat we zagen dat de hartfalen gerelateerde stollingsneiging zelfs significant verlaagd was. Ook zagen we dat een periode van 4 weken van hartfalen resulteerde in een toename in de bindweefselvorming tussen de atriale spiercellen. Dit lijkt dus aan te geven... De toename in bindweefselvorming resulteerde trouwens ook in een meer complexe geleiding in de atria. En dit lijkt dus aan te geven dat hartfalen een belangrijke rol speelt bij de ontwikkeling en de progressie van atriumfibrilleren, maar onafhankelijk is van de stollingsneiging. Ter afronding wil ik dan nog de belangrijkste bevindingen voor jullie samenvatten. Wij hebben getracht de relatie tussen de hartritmestoornis atriumfibrilleren en hypercoagulabiliteit, oftewel verhoogde stollingsneiging, te onderzoeken. In hoofdstuk 3 lieten wij zien dat fibroblasten die werden gestimuleerd met stollingsenzymen reacties gaven die kunnen leiden tot bindweefselvorming en inflammatie. In hoofdstuk 5 zagen we dat AF, dus de aanwezigheid van AF zonder andere risicofactoren, niet resulteerde in een verhoogde stollingsneiging. Wel zagen we dat AF resulteerde in vergrote atriale spiercellen. En bovendien zagen we dat behandeling van antistolling tijdens atriumfibrilleren het ontwikkelen van deze vergrote spiercellen voorkwam. In hoofdstuk 6 zagen we dat de combinatie tussen hoge leeftijd en AF resulteerde in een verhoogde stollingsneiging. En bovendien ging dit gepaard met de ontwikkeling van vergrote atriale spiercellen al na 4 weken van AF. Het laatste experimentele hoofdstuk liet zien dat hartfalen resulteerde in een verlaagde stollingsneiging, wat gepaard ging met toename van bindweefselvorming en complexere geleiding in de atria. Dus ter afronding wil ik eindigen met onze conclusie en titel. De relatie tussen atriumfibrilleren en hypercoagulabiliteit is meer dan een tweerichtingsstraat. Het blijkt namelijk een complex netwerk te zijn van vele zijwegen. Bedankt voor jullie aandacht en ik geef nu het woord terug aan de prorector.
Hartelijk dank voor deze heldere presentatie. We now switch to English again,
Correct.
if you don't mind.
Well, I have no choice.
And we start with the opposition. And the opposition will be opened by Professor Reutelingsperger, Professor of Biochemistry of Apoptosis at this university.
Thank you, Pro-Rector. Dear candidate, also my compliments to you. This for the book, for the thesis, which is very interesting and I think very valuable. Your clear presentation, and that also includes your promotion team. So this is a special occasion, 2 promotions on the same day with the same book. And that is also a challenge for us to ask some questions. There are a number of questions. I would like to start first with a question about your GOAT model. In your presentation, you already explained a little bit why you had chosen for the GOAT model in order to get knowledge about the process of AF, and in order to be able to translate your findings to the patients. But my question still remains, because I was wondering why you had chosen, you and your candidate had chosen for this GOAT model. Because there are some differences between goat and man, and in fact, if there is a xenotransplantation occurring or investigated in these days, it is the pig-to-human transplantation. And there is also a pig-to-human heart transplantation that has been carried out in 1 patient, and research is going into that direction. So my question is, why did you choose the goat and not the pig model?
Okay. Highly esteemed opponent, thank you very much for this question and your nice words. If I get your question correct, your last question was actually why we chose for the goat model instead of the pig model. And if I'm well informed, I know, or I would answer like this, because the goat model of atrial fibrillation, well, the goat is very suitable to induce atrial fibrillation. And in the pig, I think they also tried to induce atrial fibrillation, but very often this resulted in ventricular fibrillation, which worsens the outcome, of course, for the animal, and it makes it not really a suitable animal model to maintain the arrhythmia for a longer period. So that would be my answer for goat versus pig. And then your first question, that was mainly talking about species-specific differences, and indeed we encountered that as well. especially for the coagulation assays, because there are assays available that are very suitable to get nice information about specific coagulation factors that are suitable for human, but also for mice. And we found out that they were not optimal to use in the goat situation. So indeed, if I should translate the goat model to the human model, there are, of course, differences, but the development of the pathology, so the structural remodeling, electrical remodeling, that is quite translational to the human situation.
So does that imply that the goat heart is closer to the human heart than the pig heart?
Well, that can depend on many factors, but also I don't think we ever try to induce atrial fibrillation in such a way on humans, so I don't know if that would result in ventricular fibrillation like in pigs, but
okay, so it remains a challenge to translate your results into the human situation. The other question that popped up, I don't know whether you can answer that, but would it be something for the clinical scientific community to focus on the transplantation of goat hearts instead of the pig hearts?
Probably. Well, there's a very good reason why they chose for pig hearts. Actually, I'm not aware of that reason, but the goat heart could be a very good second option, in my opinion. But
that's a new field. Very good. Yeah. So that's one of the goals of your thesis, of course, is to provide basis for future research. So
Definitely.
That could be one. Now coming back to the translation again, and I think it is very difficult for us to find models in cell culture, in animals, from which we can extract results that give us a plausible insight in what happens in patients. And in your case, in your situation, you already came up with the problems there, because for the in vitro cell culture to show that there is a connection, a molecular connection between the coagulation system and cells, you use red fibroblasts.
Among others, yeah.
Yeah, and I asked your co-candidate about the contamination of the factor 10A used. And you know what her answer was on that. But if I look, there is a possibility that has not been excluded yet, is that red fibroblasts produce prothrombin. Now let's assume it produces prothrombin. What would be the experiment to demonstrate that the effect you saw is not caused by the contamination of thrombin? It's quite a difficult question.
Yeah, I will start to think out loud.
Yeah, do it.
Direct inhibition of thrombin, I would add dabigatran, for instance, to the mixture, but still, so contamination of thrombin.
Okay, but I think that's in a good direction. So the prothrombin is then secreted is then present in your cell culture, and the 10A you add makes thrombin out of it. So the rivaroxaban does not give the answer to contamination by thrombin.
Yeah, so indeed, we should have added dabigatran to the mixture to see what the direct effect was of rivaroxaban then.
Yeah. Do I have still time for a short question, or is it very short question? And that is about what also your co-candidate talked about, the egg and the chicken in this case, and you demonstrated that it's even more complex. So there is a relationship between the fibrillation and the coagulation in both directions, but there is also in between some. So how would you propose, and this is in the goat model, So we don't know whether it happens in a human. But how to get more insight in this egg-chicken relationship? What would you perform or propose to perform in the goat model to get more insight into this relationship?
Thank you for your question. So if I get your question correctly, you want me to speculate on possibilities to rule out possible sideways in the
for example,
complete loop.
Yeah.
Actually we did that already. We included animals with just atrial fibrillation without other comorbidities, and we looked at the effects that would have on hypercoagulability, so without the presence of age or other apparent comorbidities. So actually we did that already, and we added actually a couple of sideways to make it more complicated to see if the effects of atrial fibrillation would be aggravated by these possible comorbidities. So to come more to a chicken and the egg, I would continue with the model of lone atrial fibrillation then, because it makes it less complicated.
Okay, thank you very much. I'm very satisfied with your answers. Give the word back to the prorector.
Thank you very much. The opposition will be continued by Professor Wenzel, Professor of Vascular Inflammation from the University of Mainz in Germany. Welcome.
Thank you, dear prorector, dear candidate. Thank you very much for presenting your thesis. I was enjoying it very much to hear it in Dutch, and I apologize that I cannot ask you in Dutch. So thank you for accepting the questions in English. So my first question refers to proposition number 1, and I would ask the Paranymph to read it.
Thrombin and factor Xa activate cardiac fibroblasts and contribute to the process of cardiac remodeling.
Yes. So you nicely showed that the fibroblasts play a role in the inflammatory process also in your model. And I would like to ask you whether you could explain or discuss the effects of thrombin or factor Xa on inflammatory cells in your system and their role for cardiac remodeling.
Highly esteemed opponent, thank you very much for your question and also before for your nice words. Indeed, in vitro we only looked at cardiac fibroblasts and their stimulation with the coagulation factors. We didn't include any inflammatory cells, but we know that there is quite some communication between the cells that in the human body. So to be honest, we didn't investigate that. So I should investigate it to come to a nice answer now. But based on our results from our thesis, I cannot give you an answer on that.
Yes. Are you aware that there are resident macrophages in the heart?
I know that there are macrophages, but maybe you can explain me a bit what the resident...
Yes, so there is a population of myeloid cells in the heart, and it has been shown that they play a role, for example, for conductance disturbances, at least in mouse models. So I wonder whether you could speculate, because these cells are very, very important for tissue homeostasis and remodeling in general. So the question then arises whether the fibroblasts versus macrophages, are the more prominent source of, for example, inflammatory mediators, cytokines, for example, or growth factors. And it also implies the possibility that myeloid cells have a high plasticity and can undergo a phenotypical change. Yeah, so maybe you have observed something like that in your experimental work, that you came across some findings that you maybe could not really integrate at first in your concept that it's all the fibroblasts. So I was just curious to hear from you whether you could share some thoughts with us.
Thank you for your question. I know that under certain stimulation, fibroblasts can differentiate into other cell types, to be honest, in chapter 3, yeah, like I said before, we didn't investigate the influence of macrophages, but under certain stimulation, we know that cells can differentiate into other type of cells, definitely.
Okay. So I have another question. You nicely showed that heart failure promotes AFib burden, at least that's also known from human medicine. But on the other hand, AFib also increases the burden of heart failure. Yeah, so it's a mutual relationship. And can you briefly explain and discuss the effect of AFib on heart failure development in your goat model? Yeah, AFib alone on heart failure development.
Yeah, thank you for your question. In our goat model, we did it the other way around, indeed. So we induced heart failure in our goats, and then we induced AF, and we really saw that there was a strong substrate for AF induction, but also progression. The other way around, we never tested that before, but if I think about it, we know that atrial fibrillation induction leads to electrical remodeling, but also to structural remodeling. And maybe these structural remodeling changes can also contribute to remodeling changes that are important in heart failure. Also pressure changes, so that potentially can lead to a more easily inducible heart failure as well. But the way we induce heart failure is, of course, by left ventricular pacing. So, yeah, I should think of how to test that in a goat model, but how we investigated that was the other way around. So first atrial fibrillation.
So I came across that question because I read chapter 7, and in figure 2 of chapter 7, you induce heart failure, and then you put AFib on top, and then you describe the development of LV ejection fraction, and there is a little difference, you know, when you induce AF on top, you even further decrease LV ejection fraction. So that's why I wonder whether you included another control group just investigating AFib alone.
Yeah, you referred to the change in figure 2, so yes. At time point 3, indeed. Yeah, that is indeed, there is a difference of 10% that didn't turn out to be significant. But even at a later time point, we see the same direction of going downwards again. So, yeah, we didn't investigate why and how this was affected. But actually, per goat, we adjusted the pacing frequency also based on how the animals behaved on their physiological behavior. So there can be inter-goat differences, which can, be the reason for this difference in ejection fraction. But overall, the group shows a similar pattern.
Okay, thank you very much. I'm satisfied with your answers.
Thank you very much, Professor Wenzel. The opposition will now be continued by Professor Schroen. She's Professor of Experimental Cardiology at Maastricht University.
Thank you so much. Dear candidates, also my congratulations to you and to the team for this very beautiful work. And I really liked the approach you took for the presentation, which was so different from Elisa's presentation. And that may also be a very nice introduction into my question, but it may not. Because I want to talk about sex differences. And I've read your thesis, obviously, and you've had multiple chapters with the goat model. And for chapter 5 and I think 6, I could see that you used female goats. For one of the chapters, it wasn't entirely clear what you used. So can you please clarify?
Yeah, highly esteemed opponent, thank you very much for your nice words and this question. So all goats that were included in this thesis were of female sex, indeed, and we have a good reason to include females only. However, based on translational perspective, that is of course not a downside, but it makes it interesting to see if the same differences would occur in male goats. The reason why we included only female goats has actually different reasons. Practical reasons mainly. male goats are quite dominant and very difficult to house, actually, especially if you have such an intense investigation and you have to work in such close contact with goats. So female goats, that's a benefit of female goats. But next to that, also our goats come from milk farms. So these milk farms, of course, they keep the goats for their milk, and that's why only females are available, and especially for the goats in chapter 6, of course, where we included old goats, then it's very difficult to find old male goats. But again, to speculate about the differences between those sexes, we know that, there are cardiovascular differences between females and men. However, this could, for this hypothesis, that might have been a benefit as well, because we know that the stroke risk is often higher in female sex. So based on the coagulation activity that we also investigated, that would maybe even show an even earlier difference than if we only included males. But for future experiments, and especially for the whole scientific community, it would be good to include both sexes and to look at the intersex differences, of course.
Wow, so you've really gone ahead of all my questions, so that's great. So thank you for that answer. Let me think. You mentioned stroke risk being increased in women. How can I put that in relation to AF? To my question. Yes, these goats were still in the reproductive age, right? The most of them.
In the reproductive age, that's correct. But of course, they also have a certain cycle, and we didn't keep the cycle into account, but we made sure that the goats that were included, that they were not lactating anymore. But of course, to keep lactation ongoing, they need to breed every now and then, of course. But before they were included, they went through a dry-off period. So we made sure that there was no lactation ongoing anymore. But they were still in the reproductive age, definitely. Otherwise, they wouldn't be present at the farm anymore.
Yes, so I'm well aware of how that functions indeed. But could you speculate then on the lack of fibrosis that you see in your chapter 5, and could you link that to the use of female animals?
Thank you for your question. Well, we know that fibrosis due to AF occurs in men and in women, but the absence of fibrosis in our goat model, I don't think that is a consequence of the sex differences. Like I proposed already in my talk, I think it's mainly due that we made use of lone atrial fibrillation, or that our period of atrial fibrillation was too short.
But it is similar to other studies you did, right?
Our findings, you mean?
Time frame.
The time frame, more or less, of course. Yeah, there are goat models of 6 months, but also of shorter periods. And of course, it also depends on where you look for the fibrosis. For instance, we specifically look for a certain type of fibrosis, endomysial fibrosis, and that was not present in our model after 4 months, indeed.
Yes.
Flown AF.
Okay, yeah, I was relating it to the age that women on average have when they develop AF. Can you say a bit more about that in relation to your model?
Well, our goats were relatively young, so to be honest, I'm thinking now in my mind: do women get AF earlier or later than men? To be honest, I don't know. I don't have the answer, but actually the age of our goats wouldn't influence that, because our goats were relatively young and there is no spontaneous AF development in our goats.
So it's difficult to speculate on that. I can see that, yeah. Do I have time for one final question?
1 minute.
1 minute. So I would like to then quickly switch to broader effects of hypercoagulability, and you related in your discussion also to increased platelet reactivity. You didn't include platelets that much in your investigations, and also what I missed is a bit the vasculature. I do see vascular smooth muscle cells, but I don't see endothelial cells. So I was wondering if you could include those two circulating and vascular factors in your hypothesis.
Yeah, thank you. Well, they especially endothelial cells as well, they play an important role in our hypothesis because, the increased thrombogenic risk in atrial fibrillation is thought to be caused by different factors, so changes in blood flow, but also, for instance, vascular damage, so leakage, for instance, from coagulation factors to the tissue, and hypercoagulability, so the constituents in the blood. And we looked at, for instance, inflammatory markers in our gene expression that are known to be also involved, that can lead to endothelial damage. But specifically, we didn't look into that yet, but that would be very interesting to do for future experiments, and we collected lots of samples to do that indeed. Platelets, especially for the coagulation part, we had to start somewhere, and to make it not too complicated, we focus on hypercoagulability, so the secondary hemostasis, because platelets, of course, play an important role.
Yeah, you had a very good focus in your thesis, but I'm happy that you say that you will include those, and then please also involve the sex differences.
Sorry?
Please also involve the sex differences.
Yeah, I will try.
Thanks, and I'm happy to give the word back.
Thank you very much, Professor Schroen. The opposition will be continued by Professor Gutte. Professor Gutte is present online. He's professor of cardiology at St. Vincent Hospital, Paderborn, Germany. Professor Gutte.
So thanks again for the very nice presentation of your thesis. I have a question with regard to your chapter 6 and the conclusion. You pointed out that aging, or the aging process, is related to atrial structural remodeling on one hand side, but on the other hand, the complexity of atrial fibrillation in your study is not related to age. So what does it mean that the aging process changes atrial tissue, but the complexity is driven by something else? Could you explain on this?
Highly esteemed opponent, thank you very much for your kind words and for your question. Yeah, complexity indeed is determined by the level or the severity of remodeling. And we also know from clinical studies that remodeling is also age-related, of course. That can also be due to other comorbidities that are present during the aging process. And if I get your question correctly, we claim that indeed age is related to structural remodeling, but we don't see baseline differences in acutely induced AF, for instance, between the groups. Well, possible or an explanation for that could be that the age difference in our goats might not be big enough to really translate the changes, the atrial changes into complexity. Our goats had indeed an age difference of at least 6 years, but of course in humans aging takes much longer, and other comorbidities can add to the level of remodeling.
Yeah, I think that could be a very good point indeed. Another question what I do have is, interestingly, and this appears to be a bit in contrast to results we have in humans, where the presence of atrial clots, if you do a TEE study, is in particular present in patients with heart failure. And in chapter 7, you describe that in your results, the presence of heart failure reduces the clotting potential. How can you explain this? This would be one question. The second question was, have you actually seen atrial clots in your model?
Thank you very much for your question. Let me start with the second question. We didn't see clots occurring in the OR or thromboembolic events happening in the goats. Not in the heart failure model, but also never in the AF models. And the first question you were referring to, so what could be an explanation for the reduced coagulation activity or coagulation potential in the heart failure goat model? Yeah, for us that was also a very striking finding, and we came up with different possibilities. So one could be that since we saw congestion in the organs of these heart failure animals, that there can be liver damage, and the coagulation factors are synthesized in the liver, which could then reflect the fact that they indeed are hypocoagulant. But another option could also be that there is consumption of all the coagulation factors in the sample, which actually then shows on the thrombin generation that they seem hypocoagulant, but actually they are very prothrombotic. We didn't investigate that, so we can only speculate about that, but that could be two explanations going both in a different direction.
Yeah, fully agree. So the last question I do have is, since you work in vivo models, and I think it's always nice to have in vivo models because you have, of course, the differences or the changes in the heart you're investigating, but you may also have several non-cardiac condition factors also, which may have an impact. So do you think that within your model that you have non-cardiac factors which actually also had an impact on the clotting system and the results you received?
Thank you very much for this question. Definitely, indeed, in vivo models, there are many more factors involved in the outcome. For instance, when I now think about it, stress could be also of influence on the cardiovascular outcome. And these animals are, of course, social animals, and we take them from a farm. So it could be that stress also influences the cardiovascular outcome. But at least that's then it occurs in all animals. So that's a factor that's from outside the body, of course, but inside the body, probably indeed an in vivo model is very complex. So many different other processes can be involved, but I find it difficult to speculate about that right now.
Yeah. Well, one direct question is, do you think, or do you have any information about the kidney function? Because especially if you have heart failure individuals with atrial fibrillation, kidney function may deteriorate, and also then a lot of things change or are also altered due to the kidney impairment. Do you think that some of your goats developed some type of kidney disease or so?
Thank you very much for this question. Yeah, indeed, we saw some necrotic cores indeed in the kidneys, and we also, like I said, we saw liver damage or bluish livers that occurred due to heart failure. So there we see a specific link with heart failure and the outcome. But before we include the animals, of course, we make sure that all animals are more or less healthy for the level of that we can choose, of course. But we checked that there were no liver pathogenic liver values. We also checked for infections in the milk. So these factors, if we wouldn't have checked for that, they could definitely influence the outcome. But we carefully selected our animals to have a more or less comparable baseline.
Okay. Thank you so much for your answers. I'm satisfied.
Thank you, Professor Goethe. The opposition will be continued by Dr. Blankenstein. He's Associate Professor of Pharmacology at this university.
Thank you very much, dear candidate. I also would like to congratulate you and compliment you on the thesis that we are discussing. And I want to ask you a question on chapter 5. As you know, I'm a pharmacologist, so I looked at the pharmacokinetic data with interest. And I noticed that the dose that you give to your goat is actually 3 milligrams of rivaroxaban twice daily. And if I add that up, then that's 360 milligrams of rivaroxaban per day. Maybe you don't have the clinical data available, but I looked in the Pharmacotherapeutisch Kompas. And for humans, the maximum dose is twice 15 milligrams, adding to 30 milligrams per day. So in goat, you are about 12 times higher than in humans. And I would like you to comment on that. Do you have any idea what's going on there?
Esteemed opponent, very much thank you for your question and for your nice words. Indeed, we treated our goats with a high dosage of rivaroxaban. In fact, or maybe it doesn't directly answer your question, but I think we used the amount of rivaroxaban with which you could treat a human for 81 years just with that study. So you're right, it's a very high dosage. But this dosage was carefully selected based on a PK study together with the manufacturer, so biopharmaceuticals. And this dosage was based on the prothrombin time prolongation. So actually the clotting time, more or less. And then a prolongation of 20% to 50%, so 1.2 to 1.5, and that resulted in a specific therapeutic range where we based our dosage on. And why is it that much higher than in humans? That's a very good question. Well, the goat is also a different animal than a human, of course. They are ruminants, so it could be that that infected the way how they absorb or how the drug affects their works in their body. But we compared it to the clinical outcome. So, and in there is also the rivaroxaban dosage, which is more or less based on a PT prolongation of 1.2 to 1.5. So that's where we based our dosage on. And indeed, we did it 2 times a day, 3 milligrams.
Yeah. So based on your remark, you think it's an absorption problem, not so much a metabolism or elimination problem of rivaroxaban that makes you give these very high doses?
Thank you for this question. I'm now thinking, what's the difference between absorption and the excretion? To be honest, I don't know why we had to give so much more, but the outcome in the clotting time was comparable to the human situation. Yeah. So that's where it's based on. We never investigated further what's the difference. Well, actually now when I'm talking about it, I know that the protein binding in goats is completely different compared to humans. I believe it was 18% in goat and 5% or so in humans. So actually it could be in the free fraction of the drug actually where the difference is. So we never investigated that further, but we received some indications from the manufacturer.
Okay. Well, continuing on this, you gave the rivaroxaban to inhibit factor 10A, and you want to inhibit factor 10A to prevent the activation of your protease-activated receptors, right? That's right, a common theme in your different chapters. Now my question is, this activation is clear. You cleave off part of the receptor, but did you also think about inactivation of this receptor? Are there also mechanisms that can turn the receptor off again?
So direct PAR inhibition, for instance, is that where you're referring to?
Well, that's a pharmacological way, but maybe also the physiology has ways to turn off the signal that you turn on. Maybe I should explain myself. In normal receptor pharmacology, you put a compound on a receptor, but it also can come off. But in this case, you cleave off part of the receptor, and it kind of stays activated. So that's a remarkable difference.
Yeah, I have to acknowledge that that is indeed a remarkable difference, but I can't speculate about that, to be honest.
Well, maybe I can help you a little bit, because in your gene expression profiles, you looked at caveolin.
True.
Does that give you a hint on what could happen here?
Yeah, so we included caveolin as one of the target genes in our analysis because it's known that it's involved in the internalization of the PAR2 receptor. That's what I know about it, but much more I cannot speculate about it.
No, well, actually internalization is a very common mechanism to reduce receptor function. Okay, thank you very much for your answers, and I return the word to the prorector.
Thank you very much, Dr. Blankenstein. Opposition will be continued by Dr. Naji. He's postdoc at the Department of Biochemistry of this university.
Thank you. Dear candidate, first of all, I would like to congratulate you too with this nice achievement, and also to your supervisory team. So I would like to continue the discussion on basically your model. So because you chose I'm not very familiar with how to translate directly so the ages of your goats. So also how long the AFib was present. So how can you that translate to human situation? So in your case, it was weeks, months. What does it mean for actually humans?
Esteemed opponent, thank you for your nice words and the question. Yeah, of course, atrial fibrillation is a disease that develops at a certain age or at a certain moment, and all the time that the human already lived, it was exposed to different stressors. So many factors could already contribute to the substrate of atrial fibrillation. In our goat model, or in general in a model, we induce atrial fibrillation for a certain time. These animals, they also went through some stressors in their life, but it was a much shorter lifespan. And also the duration of atrial fibrillation that we induce atrial fibrillation is often much shorter than the complete duration of atrial fibrillation in humans. So that could result in differences in the severity of the remodeling. And indeed, I don't think that the level of fibrosis, for instance, that we observe in our goats is comparable to the level of fibrosis in humans. But of course, we look at a very early development, and that is where we want to find connections.
So also you aiming basically to find a way of maybe also recognizing earlier AFib, or like?
Yeah, that's true. We look at early onset and progression of atrial fibrillation. This is not a model of permanent atrial fibrillation, of course. Then we should keep the animals much longer. So we really investigated the early changes that occur due to AF induction and the early progression, indeed.
Mm-hmm. And so upon rivaroxaban treatment, you saw these nice cellular vascular changes. And there have been studies showing that, of course, AFib and stroke are really closely related. And in patients with atrial fibrillation, actually, oral anticoagulants can decrease the risk of stroke, but also decreasing dementia, vascular dementia. Do you know whether it could be the similar mechanism, so also due to, like, in that case, factor Xa inhibition, or just is completely independent?
Thank you very much for your question. I think in dementia, also microclots can be involved indeed. So atrial fibrillation potentially leading to a higher coagulation activity can indeed also influence the development of dementia, and then anticoagulation treatment could be beneficial on that. That's, if I understand your question correctly, I would answer like that.
Yeah, thank you. Thank you for the answers, and I would like to give back the word.
Thank you very much. Then we go to the last opponent, Dr. Van Eijs. He's Associate Professor at Genetics and Cell Biology at Maastricht University.
Dear candidate Billy, today is a very pleasant day, sunny day of glory, after the disastrous year of your star team, Berg 28, which sunk to the very, let's say, bottoms of the football world. Yeah, a level that we will not talk about. Any day. Today is a better day. So congratulations. Let me start with this. Do you know the long play Sgt. Pepper's Lonely Hearts Club Band? Yes?
I only heard the name of the album. I didn't hear the first word.
I'm not asking you to sing it, but...
So what was your question? Sorry.
My question?
Mm-hmm.
The question is, the first line of Sgt. Pepper's Lonely Hearts Club Band of this first song is, "It was 20 years ago today." And indeed, 20 years ago today, Victor Thijssen was defending his thesis in this same hall. And he did some differential display on sinus rhythm hearts versus AF hearts. And he came up with quite a number of genes, which he nicely fitted into categories. And he didn't find any, let's say, genes that were involved in coagulation. On the other hand, he came up with genes that were involved in structure, genes that were involved in metabolism, and so on. Now, if I compare that with the list of genes that have been looked at in this thesis, it's very different. And so my first question is, what is the rationale behind the selection of the genes that have been looked at in this thesis?
Esteemed opponent, thank you for your nice words and your nice introduction, which I didn't really understand at that moment, but now it makes sense. Thank you. Indeed, the model of atrial fibrillation, the GOAT model, is being investigated for many years already, and many things that were discovered around 20 years ago are still very important. So the reason why we selected these genes is actually based on our hypothesis. So we investigate from the hypothesis that atrial fibrillation can lead to hypercoagulability, and hypercoagulability can also contribute to the progression of atrial fibrillation. And we had to carefully select some genes because the method that we wanted to use in our analysis was PCR analysis. And to design primers for that, we found difficulties. There were many SNPs at the regions that we wanted to use or to design the primers on. So we were in contact with consortia that sequenced the whole genome of the goat, and we carefully had to select regions or genes that of interest. So we chose genes that were involved in the vasculature, in fibrosis, in inflammation, in hypertrophy, and in coagulation. So we had a select list of genes, and indeed it's not complete. Of course, if we had more time and more options, we would expand the list much more, but the genes that we included showed more or less a connection with coagulation in the literature and on the processes that we want to investigate, like fibrosis and hypertrophy.
But if I go to the sections, to the different chapters, I see that the difference, for instance, between young and old goats and young sinus goats and AF goats, most of those genes don't show any difference. So if you make a selection on the basis of what you find in the literature, there's something wrong with the literature, or there's something wrong with your selection.
Thank you for this remark. Well, some genes, indeed, we didn't see the changes that we expected. And of course, we based our choices on literature, on human studies, on mice studies, on rat studies. But actually, also some genes showed interesting differences. So, for instance, MPP-A and MPP-B that are known to be involved in heart failure and hypertrophy. They actually showed expression. So, also when atrial fibrillation compared to sinus rhythm or heart failure compared to sinus rhythm. So we cannot foresee how the genes will react, but we carefully selected them. And some indeed showed differences that we didn't expect, and others did show, confirmed our expectations.
Yeah, if I had to make a selection for something like this, I would start with a number of genes that, let's say, can be used as a control to see what's going on, genes of which I know what they're doing, and genes that, according to the literature, may be interesting. But I do not see very much of such a selection here.
Okay, well, I think MPP-A and MPP-B really showed an expression like we expected, but the selection of the pars, etc., yeah, we included that based on our hypothesis. We could not foresee how they would react in these ways.
Willy Scaf, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. Of course, we also still have to discuss the other candidates' defense, so it will take a little bit longer than normally, probably, but I request that you and your company await the results of our deliberations and our return in this room.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes. In the meantime, let's take a closer look at Maastricht University's renowned teaching method, problem-based learning. Once the pro-rector reconvenes the session, we'll tune in and continue the live stream.
Problem-based learning, or PBL. What does that mean exactly? 3 of my fellow students and I will show you around. Every week, we analyze a different case or issue together. We discuss the case and everyone can contribute different perspectives to the group discussion. If we get stuck, our tutor helps us out and suggests what we could do next. I prefer going to the library to prepare. Here, I can focus and I have quick access to books or journals that help me understand the case. Today, I can also train my stitching skills in the Skills Lab, where you can immediately put into practice what you've learned. After a day like this, I like going to the gym to clear my head and get ready for the next day.
At UM, you meet people from all around the world. Hello guys. Some of them are doing their exchange semester here, and they often say that PBL helps them learn and retain things very easily. I can understand why. It's a very active way of learning because you have to bring your own perspective to real-life cases. You have a lot of freedom to manage your time, your studies, your hobbies, and your work. Of course, that also means a lot of responsibility. Right now, for example, I'm arranging my exchange semester in Madrid. How cool is that? In this group session, we're the managers who have to allocate the resources of a real company. This is how we put into practice what we learned this morning. Studying here means being proactive and learning to plan well. Prioritizing and performing well under stress are great skills that help you develop as a person. But now, it's time to grab a coffee at the Student Service Center.
Right now, we're at the Brightlands Camelot campus. Here we can apply knowledge from lectures and tutorials in a practical setting. This helps us understand what we have learned and further develop our lab skills. Today, we're determining the amounts of cholesterol in various products. What I really like is the project periods at the end of each semester, where we complete a full research project that includes planning, collecting data, analyzing, and presenting the findings. That way we learn how research works and we're able to see what it's like to be a real scientist. After practicals, I have to write a lab report that also helps me process everything I have learned today. UM has a lot of learning spaces where you can work on your own or with other students. This evening, I'm meeting my friends for a movie night organized by the MSP Study Association.
If you study law, you have to read quite a lot. Not all information is relevant, so you learn how to easily find the information you need to solve your case. In the afternoon, I have to give a presentation, so I like to practice it with a fellow student. Later today, a lawyer is giving a lecture. This will help us better understand the case we're working on. Speaking in front of a group is quite exciting the first time round, but you get used to it quickly. And having to present helps you also to adopt knowledge better. What I like the most is that we sometimes get to enter a plea in front of a real judge. These moot courts are really exciting. Now it's time for drinks with friends. Studying is important, but so is relaxing once in a while.
boekjes door. Wil je een boekje? Of niet? Nee, ik kan het niet vasthouden. Oh ja.
We first start with Elisa D'Alessandro. Elisa D'Alessandro, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor ten Kate is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
Good. Thank you. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Elisa D'Alessandro, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Dottoressa D'Alessandro, Elisa, it's a great pleasure to introduce this laudatio that will be continued by your co-supervisor, Frans van Nieuwenhoven. But I claimed a first minute to talk to you, and I read it out because otherwise it will become 10 minutes or something. But the first thing I wanted to say is you have a beautiful suit, and it suits you very well. And well, maybe I'm not allowed to say that anymore these days, but I'm old enough to say it, I think. So consider this a compliment. But you always were dressed nice, and you always were in good spirit. And now I start reading what I really wrote. That's better, that's safer. I remember our first meeting and the positive impression you made. You looked enthusiastic and eager to start on this PhD track, and I immediately had a very positive feeling about you. And why is that? I've been working with many Italian students over the past years, and PhD students and faculty members, and I always find it a great pleasure to work with Italians. It feels like family, in a way. I mean, it's pleasant, and you're smiling, and we talk about Mediterranean food, and your grandmother cooking pasta, etc., and that makes me hungry, raise my appetite, and wanting to go to the south of Italy as well. So we join certain Mediterranean sentiments. But it's not just that. I think also as a person, you're radiating positive energy, virtually always on the bright side, even at times of major stress that you have suffered from during this PhD track, the challenges in the lab or otherwise, but you maintain a positive appearance and a good level of energy. You are a strong and persistent person that does not easily give up. This is an important characteristic for someone like you who wants to succeed in molecular biology, which is also the signature on your LinkedIn account, as I noted, molecular biologist. And you can claim that, I think, because you have all the right genes and the right phenotype to succeed there. It's with great fondness that I will think of you and the excellent, and also of you as a steering member, a driving member of the team that we have been so lucky to work with. and also so fortunate to work with Billy, of course. And I'm not going to talk to you, otherwise it will take another 5 minutes. But together you were a great team. But Elisa, yeah, I give the floor now to Frans, but I wish you a fantastic life and a fantastic career. Enjoy with your friends and family here together. And now I pass over to Frans.
Dear Dr. D'Alessandro, dear Elisa, it is with great pleasure that I speak words of praise to you today. First, I would like to congratulate you with the hard work of the past few years, the experiments, the writing of manuscripts and this beautiful thesis, and finally with the way you presented and defended your work today. Congratulations to you, to Luc, to your parents, to your friends and family in Italy and here. Uli asked me during the summer of 2017, that's a little over 5 years ago, whether I would like to join in a research project on coagulation and atrial fibrillation. And this was the RAISE 5 project that you were working on together with Billy. And you were actually the one that was especially interested in the cellular part of the work plan, and therefore you joined my small cell research group. Although I thought at first that you were a little shy when we first met, that soon changed. I found out that in fact you are an assertive and passionate person, and that we had very lively discussions during our weekly cell meetings, which I always enjoyed a lot. It was a lot of fun disagreeing with you. And then I discovered that you are hardworking and ambitious, and you soon came up with good ideas for experiments. Your experimental design was super complete, but also super complex. And I had to bring your enthusiasm in line with reality and the limitations of time and cell numbers. You performed the experiments with great accuracy, carefully analyzed the data, and had good conceptual ideas how to explain the findings and relate them to our hypothesis and research questions. You're always thinking ahead and planning for the next set of experiments or new interesting research ideas. But we had a very limited number of human cardiac fibroblasts available, so that's why you went to the research group of Neil Turner at Leeds University to verify in human cells what we had seen and discovered in the red cells. And during this period, the corona pandemic hit Europe, and both the UK and the Netherlands went into lockdown. So luckily, you were able to finish the most important part of the experiments, and you were able to return to Maastricht in time. And aside from all this work with the isolated cells, together with Billy, you continued the GOAT experiments and combined all the data finally to write the manuscripts that are published today, and the other chapters of the thesis, which hopefully will be part of future publications. Elisa, I'm really honored to have been part of your supervising team. I'm very proud of you. I'm proud of the work that you established, and I'm sure you do very well in your future career. So I wish you a bright future, or in Italian, un futuro illuminoso.
Dear Dr. D'Alessandro, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. And I hope you enjoy the rest of the day.
I will.
We will now continue with Billy Scaf. And let's see, yeah, it's difficult. Two pages of text. So I start now with your text. Billy Scaf, the degree committee here present has assessed the quality of your thesis and your defense, and in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Schotte is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan, eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Ja, dat beloof ik.
Goed zo. Krachtens de bevoegdheid, ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hierbij u, Billy Andreas Laurentius Scaf, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan overhandig ik nu de bul, door rector, secretaris en overige leden van de commissie ondertekend en met het grootzegel van de universiteit bevestigd. Geachte dokter Scaf, beste Billy, nou, ten allereerste van harte gefeliciteerd. Je hebt het heel goed gedaan. Elisa heeft het ook heel goed gedaan. Prima verdediging, heel erg stevig. Het was ook best wel een ingewikkeld onderwerp. Dat is ook duidelijk geworden bij de hele discussie tijdens het beraad, maar ook hier tijdens de verdediging. En dus gefeliciteerd. Mijn felicitaties gaan uiteraard ook naar je familie. Ik ben heel erg blij straks nog beter kennis te kunnen maken. En ja, Billy, normaal gesproken kijken we zo'n beetje terug hier bij deze gelegenheid hoe de dingen allemaal zijn gegaan. Ik heb jullie leren kennen volgens mij in het jaar 2016 of 2017. Toen hadden wij, het begeleidingsteam hier, een beurs van de Hartstichting gekregen om te kijken naar de interactie tussen boezemfibrilleren en de stolling. Best wel een ambitieus project en ook een ingewikkeld project. We wisten helemaal niet waar dit project naartoe zou gaan. Dus best wel een avontuur. Jij solliciteerde op dit project. Je was een perfecte kandidaat voor ons, want je had al ervaring met de stollingsfysiologie. Je was al een stage gelopen bij de biochemie. En je had ook ervaring met proefdieren uit je tijd in Wageningen en ook bij Charles River heb je gewerkt. Hè. Dus je was voor ons de perfecte kandidaat. En zo gingen we aan de slag. En het bleek heel erg snel, ik herinner nog goed de eerste fase van dit hele project, dat dit project allerlei voeten in de aarde heeft. En daar hebben we het ook vandaag over gehad. Stollingsmetingen die niet goed werkten in geitenmateriaal, een farmacokinetiek die heel erg anders is in geiten dan in de mens. En we wisten helemaal niet, weten ook nog steeds nog niet hoezo dat nu eigenlijk zo is. Maar toch, je bent daar op een heel erg constructieve manier mee omgegaan, vind ik. En dat heeft met een aantal eigenschappen te maken van jou. Het eerste wat ik heel erg belangrijk vind en wat ik altijd heel erg op prijs gesteld heb, is dat je heel erg open en duidelijk communiceert. Je benoemt de problemen gewoon zoals ze zijn en niet op een overdreven manier en vooral op tijd. En dat klinkt misschien triviaal, maar heel veel problemen worden groter door het feit dat ze te laat signaleerd worden en niet duidelijk genoeg benoemd worden. Nou, dat heb je dus wel gedaan en dat denk ik was heel erg essentieel voor het succes van dit project. Je bent ook heel erg goed georganiseerd. Dus we mochten altijd ervan uitgaan dat de planning goed was, dat de documentatie op orde was. Dus daar hebben we eigenlijk nooit discussies over gehad. Maar het allerbelangrijkste vond ik, Billy, is die heel erg empathische manier hoe jij omgaat met je collega's. Je bent echt een heel sociaal iemand. Je bent er altijd, je luistert altijd, je helpt altijd. Het was gewoon heel erg aangenaam om met jou te werken. Het was ook natuurlijk super aangenaam met Elisa te werken, maar ik spreek je nu even toe. En ik denk dat in je professionele toekomst, want we zullen elkaar nog tegenkomen, begrijp ik, dat heel erg nuttig is voor jou en ook voor je omgeving uiteraard. En ja, jullie hebben de problemen ook kunnen tackelen als team. Dat was denk ik ook een heel erg belangrijk reden voor succes. And this part I'm going to explain in English because I want to make sure that Elisa understands very well and that the family also understands as well. I found it absolutely amazing how you two worked together, despite the fact that you are completely different personalities. You, Billy, the Dutch guy, very well controlled and calm, and Elisa, more the outgoing, if I may say, sometimes a little bit emotional Italian young lady. But I think that worked very well together, and you had your roles in this process also. It was Elisa's part to ring the bell when things became a little bit difficult, and it was your part, Billy, to stay calm and to make sure that we have all the information together that we need in order to take a good decision. And I think I found it also very nice when I read the acknowledgement. You know, the acknowledgement is always the most important part of a thesis. Everybody reads the acknowledgement. Now, the way you comment about each other, I've liked very much. You were talking about a little bit of friction here and there. Interesting, I never saw that, but okay. That obviously happened here and there. But you had there is a high degree of mutual acknowledgement of the way you worked together. And that, I think, is very nice to see. And that will, of course, always remain, I think, in your memories and in your hearts for your entire lives. Ja, en ik ga nu weer door in het Nederlands, Billy. Je hebt hier nu ook een nieuwe uitdaging aanvaard. Je werkt nu bij CTCM. Dat is een bedrijf dat ons helpt klinische studies uit te voeren aan deze universiteit, om te kijken dat de documentatie op orde is en deze dingen. En ik weet niet of je het gelezen hebt, Billy. Jij bent straks een van die monitoren van een van mijn klinische studies. Dus hij is straks mijn baas en gaat mij op de vingers kloppen als ik iets niet ga doen. Dus dat wordt nog een heel erg interessant proces, Billy. Oké, dus nogmaals, Billy, het was een enorm genoegen om met jou te mogen werken. En nogmaals, van harte gefeliciteerd.
Dr. Scaf, ook ik wil u namens de Universiteit Maastricht van harte feliciteren met de behaalde dokterstitel. Geniet ervan en heel veel succes met je verdere carrière. En ik begrijp dat dat gewoon soepeltjes doorloopt. With this, I also would like to congratulate family, friends and colleagues from both the young doctors. Of course, I'd like to say congratulations to the promotion team, sitting on the opposite. And I'd like to thank all the members of the degree committee, and really a very special thank, because having to ask questions to two different candidates in two different sessions of 45 minutes is really a big task. So thank you all very much, especially our guests from outside, Professor Goethe and Professor Wentel. Thank you very much for being here, being online. And yes, with this, I think we end this academic session. I would like to ask the audience to leave the room on the staircase, and then we will join you in the rafter for the reception, which is the same for both candidates, I guess. So the people who want to make pictures and parents can stay here.